Welcome, everyone. This is Christy Sullivan, Secretary of the American Society for Cellular and Computational Toxicology. We're a member-supported volunteer society dedicated to advancing science and policy of in vitro and computational toxicology. And I want to welcome you to this webinar, which is co-sponsored by ASCCT and the European Society for Toxicology in Vitro. Today, we have a treat. We have 3 students presenting award-winning research. If you're a member of ASCCT or ESTIV, welcome and thank you. Please consider becoming a member to support these and other activities. I wanted to call your attention to a couple of upcoming activities. Our next webinar has just been announced. It's going to be on April 25th. And featuring some work on adverse outcome pathway identification. You can go to ascctalks.org to register for that. Also, in April, ESTIV is going to be putting on an applied in vitro toxicology course in Bucharest. There are just a few spots left. So if you're interested, go to estiv2019.com and you can find out more. And finally, save the date for September 25th and 26th of this year. We're going to have the 8th annual meeting of the ASCCT, and that will be on the NIH campus in Bethesda, and you can find in Bethesda, Maryland. You can find more about that on our website. So today, just some real quick housekeeping matters. The webinar is recorded and all attendees are muted. If you have a question, and we welcome questions from each of the presenters, please, while they're talking, add your question into the question module that you can see on the GoToWebinar control panel. We'll take questions after each talk, so don't hesitate. Go ahead and put your question in, and I'll read them to the presenter when they're finished. I wanted to tell you that today we have 3 presenters, as you can see on your screen. Astia Rizki-Safitri, Esther Wenzel, and Nicoletta Spinu. And I'm going to invite Matthew Vinken, president of ESTIV, to say a few words about the awards that they have won.
Thank you very much for the good and nice introduction, Christy. So as you heard, my name is Matthew Vinken. I'm the president of ESTIV, the European Society of Toxicology in Vitro. So we are the leading organization in Europe that tries to promote the use of 3R alternative methodologies, so not only in vitro, but also in silico toxicologies. We do this in many ways, both educationally and scientifically. We organize several events, several conferences, workshops, and so on, which already was brought up by Christy. So we have a conference every 2 years. The most recent one took place in Berlin. That was in October. And actually, the 3 presentations that you will be hearing today, that were people that actually presented during that conference and that got an award because of their presentation, either a poster award or Best Oral Presentation Awards, all under the theme of Advancing Tools for Predictive Toxicology. And I would like to take advantage of this opportunity also to advertise our next ESTIV conference that will take place in Barcelona in Spain, and that will be in June 2020. You will be hearing a lot more about that in the upcoming weeks and months. So just a brief introduction on our 3 speakers of today, as you already heard. So we are very well represented. We have speakers from different parts of the world today. So the first speaker is Astia Rizki-Safitri. She is a PhD candidate at the University of Tokyo, a candidate in PhD in bioengineering. And she will be talking about the establishment of bile duct tubular structure mimicking the intrahepatic bile duct morphogenesis for an in vitro bile recovery. So then we have a second speaker, that is Esther Wenzel from the Technical University in Braunschweig, so that's in Germany. And she will give a presentation on the generation of recombinant human anti-diphtheria toxin neutralizing antibody to replace equine sera. You will be hearing a lot about that. And then finally, we have Nicoletta Spino. She's a Marie Sklodowska-Curie Fellowship, PhD researcher under the supervision of Mark Cronin at Liverpool John Moores University in the UK. And she will be talking about the development and use of AOP networks to support assessment of organ level effects. So with this, I think we can go ahead and start with the first speaker, who will be Astia.
Great, thank you very much, Matthew. And Astia, I am turning the floor over to you.
Okay, so everything's okay, Christy? Oh, okay.
Yep.
Okay, so
your screen, go right ahead. Thank you.
Okay, so thank you for the introduction. Again, I'm Astia from the University of Tokyo. So I actually already presented this research like previous time during the ESTIF conference. So this time I'm going to present it again. So this is actually my PhD work. So in my work, I'm trying to establish the bile duct tubular structure, the culture system for the bile duct tubular structure for the in vitro bile recovery. And in this case, I'm trying to mimic the intrahepatic bile duct morphogenesis. Okay, so as a background of this research, so let's start with the liver. So everyone knows about the liver. So liver is actually plays an important role for many things, and one of them is for the detoxification process. So that's why this organ is always become the main target for the drug metabolisms assay or something like that. So usually drugs when enter the body will be metabolized by the liver, and liver will secrete as a secondary metabolite. We know it as a bile. So usually bile, they will record like what happened inside the body and what happened during the metabolism process. So that's why this is really important. But the thing is, like bile itself, how it produced and also transported like in the liver and outside the liver. So that's again, we may take a look like the histology of the liver itself. So liver consists of many liver lobules, like this picture, and then it's surrounded by the portal triad. And then basically the bile itself is produced by the hepatocyte, which is the main cells in the liver. And after that, after hepatocytes secreted the bile, and bile will be transported and accumulated by this particular tissue. We know it as a bile duct. So bile duct is the only tissue which is responsible for the bile accumulation. So the thing is, for the bile or metabolite assay for the drug test, we usually use the animal model, but animal model has some limitation, like especially for the human-related drug. So recently, like most of the research, they switch from using the animal model into the in vitro model or even liver on chip. But again, this kind of model facing some limitation. For example, like because mainly focus only on hepatocytes. So for the bile recovery, we need to consider like the existence of the bile duct, and bile duct itself is consists of the biliary epithelial cells. And to make a bile duct, actually we need a lot of work, because this is a kind of like a complex structure. So we need to make it in this kind of tubular structure. So the thing is, because it has a complex structure, so the simple model agriculture is it doesn't represent the whole liver, the bile duct function. And the recent research, they successfully developed a mini bile duct like in this picture. But the thing is, again, because the structure is not really representative, we need a more like a channel structure so they can deliver the bile duct. And also it's always developed inside the extracellular matrix, so it's isolated culture system. So we cannot co-culture with hepatocytes. So we cannot explain how the relation between the bile duct and also with hepatocyte, which means that there is no metabolite bile recovery. Okay, so the objective of the research. So we try to develop the bile duct tubular structure, which applicable for the in vitro bile collection. And we use this approach. So actually, there are so many types of the bile duct, but we are focusing on a bile duct which directly connected to hepatocyte, which is intrahepatic bile duct. So intrahepatic bile duct is always grow along the portal vein. So, and during the morphogenesis, it's always started with the 3 stacks of cells. So the first one is mesenchymal cells, and then ductal plate, which consists of just only 1 layer of bags, and also hepatoblast on the upper layer. So when these 3 cells stack together, so that's going to activate some of the signaling. So the mesenchymal cells, they will secrete this JAK1, which activate the NOC2. This is the tubulogenesis signals. And then the hepatoblast on the upper layer, they will differentiate into the bile duct, and they will make this kind of structure, like tubular structure. So what we are doing, so actually we try to mimic this kind of situation by stacking these 3 kind of cells, except in this case, we change the mesenchymal cells into the, we use mouse feeder cells. And we put the extracellular matrix on the top, and we culture on the PDMS oxygen permeable plate. Okay, so the first thing we need to do is we try to find the optimum condition for this kind of culture system. So we modulate 2 conditions in this case. So the first one is we modulate the ECM mixture, and the second one we try to modulate the combination of the cells. And then after that, we observe like the establishment of the bile duct within 35 days. And here's the map of the abundance of the bile duct-like structure per, yeah, that's calculated surface area per millimeter square. And we can see that here again, so the mixture of the ECM with the high concentration of matrigel is still preferable in this case. And also the combination of a triple co-culture, which means like when we mix MEF, BEC, and also hepatoblast is also the most preferable condition. And then actually in this kind of modulation, we found 2 kind of structure. So we found a cis structure, which is the most common structure, and also the partial tubular structure. So the interesting part from this structure is like we mostly find the cis structure in only containing hepatoblast culture, while the bile duct tubular structure mostly find in a co-culture system, and they usually elongated and grow along the mesenchymal cells and also back. So there's a tendency here, like these non-parenchymal cells, they support the elongation of the bile duct-like structure. And also to confirm the differentiation speed, so we assess these 3 kind of markers. So as you know, there's the alpha fetoprotein for the liver progenitor cells, CK19, that's really specific for the bile duct or biliary cells, and also albumin for the hepatocyte. And after we culture for several days, we can see like the decrease of the expression of the alpha fetoprotein, and we can see that there's the expression of the CK19 is increased during the culture system, which this is mainly support that the differentiation is toward the biliary cells. So here we may find that the matrigel rich ECM and triple co-culture in combination, they increase the bile duct differentiation and also support tubular structure. And also the next step, we need to record the tubulogenesis process. So we record at day 5, 11, and 17. And we can see here the video. You can see, yeah, okay. So we can see the tubulogenesis process during the culture day. And we also confirm the lumens formation. So here you can check in this video, Like it's at day 5 we started with the mold, like a mold like structure, and then this kind of structure will getting bigger and they will develop the lumen and they will grow towards the matrix, as you can see in the picture. So as I previously mentioned, so as I previously mentioned, there are two kind of gene which is really important for the tubulogenesis. They are JAK1 and also NOH2, and also we assess the extra gene expression. Here we use the Claudin 15. So Claudin 15 is reported important for the intrahepatic biliary duct organization. So here we can see the expression that the JAK1 is highly increased in a triple co-culture. So we use the embedded ECM as a control culture. And then after the JAK1 expression is rising, and after that NOH2 expression will rise. So that's to confirm like what happened inside the intrahepatic biliary duct inside the body, that for tubulogenesis they triggers JAK1 first and then followed by the NOH2. And here we can see that the expression of the Claudin 15 in a triple co-culture is already increased since the day 5 if compared to the embedded ECM culture. So here we can see that the tubulogenesis process is probably already determined prior to the differentiation. So this is most probably because of the supporting cells like mesenchymal cells and also the biliary epithelial cells. Okay, so the thing is like because this bile duct is a secretive tissue, so the polarity and also function is essential. So here we did some immunostaining using several markers. For example, like in these cases we use vimentin. This is for the mesenchymal cells marker, and also CK19 for the bile duct marker. And we use the tight Janssen marker and also the anion exchange. So here we can see there's a fine expression of the CK19 and a little bit expressing the vimentin. And we can see like the tubular structure also configure a nice tight junction, and there's a fine distribution of the anion exchange on the apical side. So so we also calculate the gene expression for this. So we can see that the expression of the vimentin is increased after day 11. So here's this phenomenon is so actually the increasing of the vimentin is equal at same time like the JAK1 actually. So there's probably mesenchymal cells in this culture, they secreted the JAK1. But we can see here, we can see also that our bile duct structure also expressing a little bit of vimentin, and there is no so much different for the tight Janssen expression. And we can see that in after day 17, bile duct, they expressing the aquaporin and also the anion exchange after day 23. And we also visualize the bile analog accumulation by measuring the multi-drug resistant receptor 1, MDR1 here. So we use the rhodamine 123. So we add rhodamine 123 in the presence of the verapamil or in the absence of verapamil. So verapamil is a drug which blocks the function of the MDR1. So here we can see that in the absence of verapamil, so there's the accumulation of rhodamine inside the lumen, while after we add verapamil, so we can see like it's like really low fluorescence inside the lumen of our bile duct structure. So that means this verapamil blocking the function of the MDR1. Okay. So the next step is we because our main goal is we try to recover the bile acid from the culture system. So it's really important to combine our bile duct structure with hepatocytes. So here we combine using the culture insert. So we use the collagen membrane culture insert, and we arrange our culture, just like in the picture. So we put hepatocyte on the bottom of the compartment, and we put the bile duct tubular structure on the upper compartment. And for the assays, we use the FDA. So FDA actually, we use this because FDA is colorless, and if we add it into the cells, and cells will hydrolyze FDA into a fluorescent which has a green color. So it's really good for the metabolite visualization. And then in this case, we add FDA on the lower compartment on hepatocytes. So we're hoping that hepatocyte will hydrolyze this FDA, and then this hepatocyte will produce fluorescence, which later on accumulated by the bile duct. So what we're doing is, this is the result. So we incubate the culture for 70 minutes, and after that we remove the FDA from the lower compartment, and we keep continuing incubate the culture system. So we can see that there is no accumulation on the bile duct on the upper compartment after 70 minutes. But after we remove the FDA and we continue the incubation, we can see like there's a slight fluorescent colors on the top, on the bile duct tubular structure on the upper compartment. So to confirm this kind of accumulation, so we also calculate the fluorescent accumulation inside the tissue. So here's the result, and we can see like we compare with the monolayer bag, and we can see like there's a high accumulation of the fluorescence inside the bile duct structure, even though after washing, like the fluorescence still stay inside the culture. Okay, so from this research, so we are successfully enhanced the in vitro bile duct structure, new kind of like a new culture system for the bile duct structure, and also we're the first time capable to simulate the bile duct morphogenesis in vitro, and we also successfully demonstrate the metabolite bile recovery in vitro. So for the prospect, there's a lot. I think there's a lot of prospect for this bile duct in vitro establishment, since like because mostly people always focusing on hepatocytes. So it is really important to be able to develop the bile duct network in vitro, because I think that's really helpful for the in vitro bile duct recovery, and also that's also really helpful for the liver disease model, especially related to the bile duct or bile accumulation, or something related to like a bile duct cancer, something like that. So I would like to thanks to all my laboratory member who helped me, and also another professor, and also I would like to thanks to Steve to yeah, like gave me some opportunity like last year to do this kind of presentation. So that's all my presentation. Thank you.
Thank you very much, Astia. That was a wonderful outline of your work. We have one question already. Again, if you have questions for Astia, you can type them in the question box, and we'll read them out. The first one is, what are your thoughts on inclusion of oval cells in the model?
Okay. So the thing is, because my model, I'm using the liver progenitor cells, which is similar to oval cells, because they have like multipotency. So I think because I'm using the liver progenitor cells, so that's why I didn't use oval cells.
Okay.
But if that's possible, that would be really great, because oval cells is like oval cells is connecting between hepatocyte and also the bile duct. That will be great.
Okay. And sort of maybe a basic question, wondering, is this a human cell model or another species?
Okay, so in my case, I'm using the rat cells, because this is just more because of the technical things, because they grow faster.
Okay.
Yeah.
All right, another question. I was wondering whether you could elaborate on the cell source. Okay, we just did that, species. And are they primary or are they, yeah, I think they're primary, right?
Yeah, I'm doing primary cells.
Yeah, and okay, yep, great. Okay, those are all the questions I have so far.
Okay, thank you.
Well, thank you again for your presentation. Oh, I have one more question. Sorry about that. Just one more question.
Okay, no problem.
Have you been able to measure markers of disease such as GGT or ALP?
Oh yeah. So I don't do that yet, but I think I'm going to do that in the future.
Okay. Aims for the future. Good. Yeah, that's excellent. Okay. Well, thank you again. We're going to move on now to the next presenter, who is Esther Wenzel. And so, Esther, I'm going to go ahead and make you the presenter. And you'll need to unmute your microphone. There you go.
Yeah. Hello, everyone.
All right, go right ahead.
Okay, thank you very much. And hello, everyone. Thank you for the chance to present my PhD work in this webinar. My name is Esther Wenzel, and I'm from the University of Technology in Braunschweig. And the topic I'm talking now is the development of recombinant human anti-diphtheria toxin neutralizing antibodies to replace equine serum. So in the next 20, sorry. No, okay. In the next 20 minutes, I will tell you first something about antibodies in general, how they are normally generated with hybridoma technology, and how the in vitro alternative phage display is working. Second, I will give you a short overview about diphtheria, the disease, and the current therapy. And finally, I will present you the amazing results coming out from my project. So let's first start with the antibodies. So what are they normally used for? So most researchers use antibodies for their basic research. Like you heard from Astia before, for example, you can immunostain tissue or cells for identifying different cell types in a, for example, multicellular system. But you can also do diagnostic tests with antibodies. So for example, doing an ELISA. But you can also use antibodies as therapeutics, for example, for cancer or autoimmune diseases as monoclonal antibodies, or polyclonal serums for anti-snake, anti-venom intoxication, or tetanus or diphtheria therapy. So now I would like to explain you how antibodies are generated nowadays. So normally, what we can buy, so commercially available antibodies, are generated with hybridoma technology. Therefore, an animal, here a mouse or a rabbit, is immunized with the antigen the antibody should target. Then the antibody produces, the animal produces the antibody. And then the antibodies are selected either as monoclonal antibodies via B-cell extraction and hybridoma generation, or as polyclonal antibodies where the serum of the animals is purified. In the end, you get a vial with the antibody that is your mouse or rabbit your target antibody. So an alternative to this in vivo system is the in vitro antibody generation method phage display. Here, in this method, we take the variable part of an antibody, amplify the gene, and clone it into a phage with the help of a phage vector. The phage then displays the antibody on its surface so that the phenotype and the genotype are coupled. So we don't do this not only for one antibody, we do this for billions of antibodies, so that we receive in the end our human antibody phage library. To select now antibodies against a specific target, we need our human antibody gene library and give it into a well so that they are in solution. We are immobilizing the target, so the antigen we would raise antibodies against on the surface of a well. So phage presents an antibody that are against this target will bind to that target. Other antibodies we will wash away. Afterwards, the left antibodies will be analyzed as monoclonal antibodies in their ability to bind, for example, in a binding ELISA, or we make a functional assay, for example, a neutralization assay in the end. So with this technique, the phage display, we can select antibodies against nearly every target without the need of any animal. And these selected antibodies can we now engineer and produce in different formats. Therefore, we developed in our lab a kind of vector cassette system. So normally we are producing first really small formats, so soluble scFvs for making first binding tests. But for further testing, we've used this to an FC part, so to have an IgG-like format, the so-called scFv-FC format. We can then model or adjust the FC part for our needs. So we can fuse a human FC part to these antibodies, a mouse FC part, or a rabbit FC part to the antibodies. And we can also go directly into IgG format with our antibodies. So, and all of these antibodies can be transiently expressed in mammalian cell culture. So also without any need, no need for animals. So, but now let's come to the topic of my PhD work and how this in vitro generated antibodies can help to cure diphtheria. So first, some basic information about diphtheria. So diphtheria is an infectious disease mainly caused by Corynebacterium diphtheria. The symptoms vary a lot, but diphtheria is mainly known for this gray patch in the throat or swollen lymph nodes, the so-called bull neck appearance. Case fatality rate for diphtheria is 5 to 10%. But for children younger than 5 and adults older than 40, this case fatality rate can increase up to 20%. These severe effects are caused by the produced Diptheria toxin. Diptheria toxin is a typical AB toxin. So we have an A fragment that contains the catalytic domain that inhibits the protein synthesis. And we have a B fragment that contains two domains, the receptor binding domain, so the domain where the toxin is binding to the cell surface, and the translocational domain that leads the catalytic part of the toxin into the cytoplasm. The diphtheria toxin is a very potent toxin, so the lethal dose of diphtheria toxin is just 100 to 150 nanogram per kilogram body weight. So in the US, there are nearly no reported cases nowadays. But it wasn't always like that. Beginning of the 20th century, millions of cases were reported. So the turnaround came in 1923 with the global diphtheria vaccination. Nowadays, we have a vaccination coverage globally of 85%. With good vaccination coverage, for example, in North America, Central Europe, or East Asia, but also less vaccinated areas than Central America, Africa, or parts of Asia. Despite the good vaccination coverage, there are still outbreaks worldwide for a variety of reasons. So, for example, since 2016, we have a large outbreak in Venezuela with up to now nearly 2,500 reported cases. We had last year in Bangladesh a huge outbreak among the Rohingya refugees with nearly 4,000 cases. And we had also last year in the Yemen outbreak with with over 1,500 cases. But also in Europe, we had over 300 cases over the last 10 years. And here also 2 fatalities have occurred. And these 2 deaths were totally avoidable and just occurred because the needed diphtheria antitoxin were not available in whole Europe. But now let me explain how today's therapy of diphtheria is working. So beside the antibiotic, you need to cure against the Corynebacterium diphtheria, an antitoxin is needed to neutralize the produced toxin. And this antitoxin is an equine serum. So that means that horses were immunized with the diphtheria toxin and produces the antibodies against diphtheria toxin. The equine serum itself then is the antitoxin. So disadvantages of this equine serum are obvious. It can cause serum sickness, so a hyperimmune reaction when injecting an animal product to a human. I already showed you that availability is a big issue nowadays, and also stability because it's an animal serum. Then batch to quality and variation of batches. So you can imagine every time the diphtheria antitoxin runs out, a new immunization has to be done. So it's every time a new product that's coming out from an immunization. And of course, the production in horses, production is in horses. So these conditions are not following animal welfare rules. And this therapy with the equine serum is the same therapy than more than 100 years now. So, and that's where the motivation for my project comes from. So in my project, the aim was to develop a totally human antitoxin product to replace the polyclonal equine serum. So we developed an anti-diphtheria toxin antibodies with phage display. After selection, we screened over 600 monoclonal antibodies derived from our human antibody gene libraries. Over 400 monoclonal antibodies binds to diphtheria toxin, and we found 290 monoclonal antibodies who were neutralizing diphtheria toxin in vitro. Following, I will show you the results of the characterization of these neutralizing antibodies. So the first question that came in our mind was, which domain do these neutralizing antibodies target? So why are they neutralizing? So we made a test against the different domains and selected 61 lead candidates for testing. And we could show that most of our antibodies that are neutralizing targets the receptor binding domain. So 21 of our antibodies are against the receptor binding domain, and 16 of our neutralizing antibodies targets the catalytic domain of the diphtheria toxin. So after we knew that, we analyzed the binding to the native diphtheria toxin. So how it will occur in the body when the Corynebacterium is producing the toxin. And we tested this in a titration ELISA, and we could see that they have a really good half maximal effective binding concentration of 20 to 50 nanogram per milliliter. And they showed no unspecific reaction to BSA in our example. So, but The most important assay now is our neutralization assay. It's a virocell-based neutralization assay. On the y-axis, you see the mitochondrial activity displayed. As higher the signal, as healthier are the cells. The green dotted line here indicates the untreated cells, so the maximum OD that can occur in this assay. The x-axis show the antibody concentration. In our setup, we say that an antibody is neutralizing when it reaches over the 50% OD value. So at a constant diphtheria toxin concentration, and then they are considered as neutralizers. So here are an example of 5 antibodies, and what you see is so the toxin concentration is constant on each of these curves. So as less antibody you need to neutralize the same toxin concentration, as good an antibody is. So the antibodies here more shifted to the right are better neutralizers than the antibodies more shifted to the left. So to bring this in a more compact format, I show the neutralization capacity of all our selected lead candidates in a bar chart. Here we converted the effective dose to international units per milligram of antibody with the first WHO international standard for diphtheria antitoxin. And what you can see here is in black as a reference the equine diphtheria antitoxin, and colored all the antibodies we analyzed. So you can see that in average, antibodies targeting the receptor binding domain have a higher neutralization potency than antibodies, for example, against the catalytic domain. So, and you see that we also developed some antibodies that have also higher neutralization capacity than the equine diphtheria antitoxin in vitro. So our best antibody, indicated here with the arrow, showed the 10 times better neutralization than today's therapeutic equine diphtheria antitoxin. So we have these amazing neutralizing antibodies, monoclonal antibodies, but our aim was also to combine different antibodies to mimic the polyclonal serum with a sequence-defined oligoclonal cocktail. So what we did was we hired the toxin concentration to a limit of cytopathic dose 100. That's a dose that's also used for in vivo experiments. And we tested the best 3 neutralizing antibodies against each domain. So one against the translocational domain, one against the cytopathic domain, and one against the receptor binding domain. And as a reference, we used an equine serum, so a polyclonal mixture. So what you now see is that at this toxin concentration, we have no neutralizing effect at all with the monoclonal antibodies. But when we now combine the antibodies, we can reach a neutralization potency as good as the one of the polyclonal serum. So all these antibodies are combinations of the monoclonal antibodies we tested before. So, to sum the last 20 minutes up, during my project, we overcome all disadvantages that an equine serum has. So, we developed a fully human product that is lyophilizable and stable. I didn't show the data because of time issues. We have a recombinant and sequence-defined product that can be produced in a cell culture, so in mammalian cells, without the need of any animal. So All of this work were not possible without the help of a lot of people. So I would like to thank all my lab members, but especially Michael, Stefan, and Saskia from our Department of Biotechnology. I would like to thank Paul and Tia from the National Institute of Biological Standards and Control in London. I would like to thank Jeff from Peter International Science Consortium and Andrula from Public Health England. I would like to thank my founders, Peter International Science Consortium and the Center for Contemporary Equine Studies. I would like to thank Christy and Matthew, so ASCCT and SDIF, for the opportunity to present my results here during this webinar. I would like to thank the Dürrenkamp Setbinder Foundation, who honored me at SDIF 2018 with the Best Poster Award for my PhD work. And I would like to thank you for listening and your attention. Thank you.
Great, thank you. Thank you so much, Esther. That was a wonderful presentation. While we wait for people to enter questions, I'm going to. So what are your next steps? I mean, in terms of your research and you are yourself, are you finishing up and graduating soon?
Sorry, can you please repeat the question?
Just wondering what the next steps are for your work.
Yeah, so next steps are so the combination results I showed you were really fresh results. So we at the moment confirming these with different toxin doses and doing then also tests in the direction of testing combinations and which are also the effects that occurs that combining toxins give us such a good outcome in neutralization. So these are the next tests, and yeah.
Okay. And sort of along those lines, we have a question from the attendees. What is the cost comparison for the polyclonal horse-derived antibody versus the monoclonal antibodies that would replace the animal-derived antibody? Do you know anything about that?
That's a really, really good question that I cannot answer in detail because producing these polyclonal horse serum, that is in the moment, so we have no facility in Europe where we can ask what's the cost. And I don't know if there is still one in the US left, but most of the companies that are producing at the moment this serum are in India, and we have no idea at all what are their costs.
Okay.
So we cannot say what's the comparison, of the prices. Yeah.
Okay. All right. Any more questions for Esther? I don't see any coming through. So thank you again for your presentation and for your work, indeed. And I'm going to go ahead and turn it over to our final presenter. So Nicoletta, I'm going to make you presenter now. Okay. Yeah. Yep, I can see your slides and I can hear you, so go right ahead. Thank you.
Hello, everyone. It is a great pleasure to participate today at this webinar. I'll be talking about the development and use of the adverse outcome pathway network as a concept to support the assessment of organ-level effects. It was so interesting to listen to the presentations of Astia and Esther, especially because their work on mechanistic toxicology can support the development of individual LPs. As Matthew and Christy mentioned, I'm a Marie Curie Fellow at Liverpool John Moores University, and I'm representing the N3 project. We aim to synergistically develop in vitro and in silico methods to human chemicals a nanoparticle safety assessment. So the M3 stays for the integration of in vitro and in silico tools. I would highly encourage you to follow us on Twitter, but also to check regularly our webpage. Well, the LOP network as a concept can be defined as an assembly of at least two individual LOPs that would share at least one common element, represented by a key event, including molecular initiating event and adverse outcome. Within the network, the key events will be represented by the node, while the key event relationships will be represented by an edge. As we all know, the individual LOPs are linear constructions, and usually they're an oversimplification of the biological interactions. Therefore, by using the concept of LOP network, we can better represent and better understand the complex of these biological systems, especially by summarizing into tractable components that would be suitable to be tested in vitro and in silico. And comparing to biological networks such as protein-protein interactions, the LOP network can focus not only on cellular level, but also tissue, organism level. Currently, there are 9 LOP networks that have been derived for different applications, usually for rodents and other species, but not for humans. Therefore, the aim of the research and today's presentation is to describe the way we develop the LOP networks with a focus on the study organs of the M3 project, which are brain, lung, liver, and kidney. We followed the findings that were recently published by DATMAB, and further on, we analyzed them analytically in order to identify critical paths that could support, for example, quantitative modeling. Our methodology consisted of a 4-step process. We defined several general design principles that could be further applied to any other question of interest. So we started by defining the applicability domain, which in our case, it was the study organs of the M3 project. We were interested in identifying existing individual LOPs that are known to be applicable to Homo sapiens, but also in evaluating the relationships between key events, if, for example, the key event upstream will lead directly or indirectly to the key event downstream, and also the level of the weight of evidence. The third step consisted of inspecting manually the OECD LOP wiki knowledge base, the repository of the individual LOPs, to extract manually the relevant LOPs, and then data curation, in order to avoid especially the ontology issue, and to be able to catch as many as possible key events in a network. And the final step was to generate the LOP networks using Cytoscape software tool. Would help us in identifying critical paths across each individual network to be developed for the quality. Therefore, however, was for neurotoxicity, hepatotoxicity, and one network. The boxes that are in green represent the molecular initiating events. The boxes that are in red represent the adverse outcome. And the boxes that are in blue are the critical paths identified for each individual LOP network. The key events, the boxes of the key events that are filled in color, are the most well-connected key events across the network. And we can see that for the LOP network on hepatotoxicity, the one on the left-hand side, is actually represented by a molecular initiating event, and not by a key event at cellular or tissue level. But let me explain on the LOP network derived for neurotoxicity. Here it was possible to link 9 LOPs, 9 individual LOPs, into 3 adverse outcomes, which were Parkinson's disease through motor deficits, neurodegeneration, and impairment of learning and memory, and the decrease in cognitive function. We can also notice that there are molecular initiating events that link multiple OPs. The level of evidence was annotated as H for high, M for medium, and low, and L for low. And this piece of information is really important, especially if you think in terms of regulatory application. We might be more interested in those key events that are known to have a high level of evidence. The network itself was developed using solely adjacent relationships between two key events. And with one exception, if we analyze the mitochondrial dysfunction, the link between mitochondrial dysfunction and cell injury death, the relationship was non-adjacent. And we aimed here to include the ALP ID38 as well, and to identify any other pathways that would lead as well to Parkinson's disease. But this is just a graphical representation that describes the network. In order to get a higher confidence into analyzing all the key events, it is useful to apply several metrics from graph theory and science of network. And therefore, we applied the most used analytical parameters in order to analyze this network, for example. And I'll start by talking about the level of degree, which is defined as the number of key event relationships linked to a key event. It basically gives us information about the connectivity of a key event across the network, but also about the convergency and divergency of a key event in a network. Convergency means that the key event of interest is linked to more upstream than downstream key events, while divergency means that the key event of interest is linked to more downstream key events than upstream key events. And here we can see the level of degree for the molecular initiating events of the ALP network for neurotoxicity. As the graphical representation showed us, there were several molecular initiating events that linked to ALPs. And they are formation of protein adduct, binding of antagonists to MDA receptors, and the inhibition of NIC. In terms of adverse outcomes for the same network, we've noticed that the adverse outcome of impairment of learning and memory and the decrease in cognitive function linked 7 ALPs. Neurodegeneration has a double role. So this can be found as a key event for some ALPs, but also as an adverse outcome for one ALP. While Parkinson motor deficits linked only one ALP, as so far in the ALP wiki knowledge base, there is only one ALP developed for this endpoint. Here we can see the overall connectivity of all the key events across the network. The most well-connected key event is the cell injury death. It is followed by the impairment of learning and memory as adverse outcome. And usually the least connected key events across the ALP network are represented by the molecular initiating events. But in our case, we've noticed the same result for the Parkinson motor deficits. Another interesting aspect of this analysis is the identification of convergent and divergent key events, which was possible in our case to do. And this piece of information can be used further into grouping all these key events and to think about a battery of in vitro tests, for example, or developing in vitro assays that would cover multiple key events in order to evaluate the final endpoint. An interesting analytical parameter is the eccentricity. It is basically a parameter that measures the most upstream and downstream key events. So usually the most upstream key events would be molecular initiating events, while the downstream key events will be the adverse outcomes. We can see that based on the scores for the eccentricity, one of the critical paths that was identified was the reduction of BDNF. and that would lead further to the cell injury death. Another interesting parameter is between centrality, and this measures the number of the shortest path between two key events across the network. And these results confirms that the critical path, the most centrally located key event, is the cell injury death. And through this cell injury death, we can see that actually it is involved in several signaling pathways. So different LPs will lead to this key event, for example, the reduction of BDNF or mitochondrial dysfunction, or the increase of pro-inflammatory mediators. But at the same time, cell injury death will lead to different adverse outcomes. For example, through neuroinflammation and degeneration of dopaminergic neurons of nigrostriatal pathway, it will lead to Parkinson motor deficits. While through the neurodegeneration at hippocampus and cortex level, it will lead to impairment of learning and memory and decrease in cognitive function. If to analyze the same network for the path occurrence, which is a variation of between a centrality, we can confirm once again the results of the most well-connected key event, but also identifying other well-connected key events across the network. Basically, it also measures the shortest path, but usually between the MIEs and adverse outcomes. To sum up, even though the OECD LP wiki knowledge base is not so rich in individual LPs, and the majority of LPs are under development, we still can derive LP networks with different applications. In our case, we were interested in developing LP networks that would be applicable to Homo sapiens and especially to the study organs of the N3 project. And 5 LP networks were possible to be generated using Cytoscape software. We then applied several analytical parameters in order to identify critical key events across each LP network. I showcased today the LP network derived for neurotoxicity simply because it was the richest in key events. But these results can be further used to prioritize the key events in terms of testing of the development of in vitro assays, creation of a battery of in vitro test methods, for omics analysis, radar crossing. We also use these results for the selection of the chemicals which to be tested within the N3 project for their toxic effects, while in my case, I'm using these results for further quantitative modeling of LPs. I would like to acknowledge my research group from Liverpool, the Chemoinformatics Research Group, especially Professor Mark Cronin, Professor Judith Madden, and Dr. Stephen Enoch. I would like also to acknowledge Joint Research Center from Italy, Dr. Annabelle Price, Dr. Brigitte Landisman, and Dr. Andrew Orff for their feedback on this work, the N3 project and all my PhD colleagues and PIs, as well as Steve for the award and for the support that it gives to PhD students. Thank you very much for the attention, and I'll be happy to take any questions.
Great, thank you. Thank you, Nicoletta. Matthew, you had a question?
Yeah, if I'm allowed to, Nicoletta, very much, well, thank you, I would say. Thank you so much for this very nice presentation. I'm always impressed by the work of this, very interesting project. But I was wondering, maybe I missed this, but how do you deal during the development of the AOP networks with homeostatic adaptations and feedback and feed-forward loops?
Thank you, Matthew, for the question. Indeed, this is really an important aspect that we need to keep in mind. And I would say, based on my experience, in the OECD LP wiki knowledge base, this piece of information is not included. And currently we have this kind of recovery mechanistic knowledge included only on hepatotoxicity. It's actually the work published under a name. And we hope that within the N3 project, we will be able to add with expertise and in this area, and then we can model in silico as well these adaptation processes. So right now, indeed, the results are based on the information taken from the OECD AOP Wiki knowledge base. So it's not enriched with literature review or some other methods.
Okay, thank you so much.
I have a question as well. I was wondering, what is, I guess the AOP knowledge base is an ever-growing resource, and what is one thing that you think would improve your ability to make these networks and gain information from them? What is missing, I guess, from the AOP knowledge base?
Well, that's also a good question, because indeed, while developing the AOP networks, it happened to have several issues. One of them was related to the ontology issue. So it is still necessary to harmonize all the terminology. Mm-hmm. And another aspect is actually, whenever you want to develop a network, it's really great to get in touch with people that develop that network, because there are many other aspects that might not be included in the page description of the individual AOP. Mm-hmm. And I still would encourage that even though the individual AOP is not approved by the OECD, but it's rather under development process, I will still encourage to use it. And at a later stage, you can evaluate those paths of uncertainty, for example.
Yeah, I think that's an excellent point. We have one question from the attendees. And again, if any other attendees have a question, you can either type it into the question box. You can also raise your hand if you want to do that, and I can try to call on you. So with AOP, AI, which is artificial intelligence and machine learning coming, how far away do you think is a major revolution in life sciences? So sort of a big picture question.
Well, I'm very optimistic, I have to say. And I'm still learning as well on how I could use and better incorporate these methods into the research field, into my research field. I believe it depends on the data availability and the way you develop the model. It should be a transparent way so that everyone can evaluate, especially the uncertainty of the model. I personally believe that there is a room for using AI and data science in our field.
Agreed, but very good points that you make. Okay, do we have any more questions? I don't see any coming in. So, all right, I think I'm going to thank all of our presenters so much for preparing their presentations and taking the time to present them. And thanks to all the attendees. Thanks to, what I notice when seeing these presentations is just how many organizations and institutes are supporting all of this work. I think it's a nice demonstration of the diversity of support for lots of different kinds of work in vitro and computational toxicology. So thanks for that, everyone in general. And again, please contact me or Matthew by visiting our website and finding contact information if you have any questions or want to engage further. And I wish everyone a good day, and thanks again for your attention. Bye-bye.
Goodbye. Thank you, Christy.
Bye, thank you.